Okay, so here is Neuroglancer that we're using, which is a version called Spelunker. And it kind of just has a few, uh, easier like tools and layout from like the base Neuroglancer that's from Google. And a lot of them we're making them backwards compatible to work with base Neuroglancer, just so you know from your experience with it. So we have our EM on the left here and our 3D model reconstruction on the right. And we have a nice, beautiful starburst here that's been reconstructed. Um, and for each of these, they correlate with these layer tabs up here. So this layer tab here is with our EM image. So if we left click it, it shows hide it. And the same goes with the segmentation layer here. That is the coloring in our 2D and also our 3D model here. You'll notice when I left click, it does show hide it. So to select that particular tab, you're going to right click. And that leads into my kind of main hot tip that I always tell anyone is if you're trying to do anything in Neuroglancer and it's not working, try right clicking or control clicking, because that seems to be like just one of the main modes that's used for Neuroglancer as an alternate thing. I'll go through the basic navigation tools here. So left click and drag is just going to rotate you around a point, and that point will be your axes. So A for axes, and that will show hide it. So you'll see it's appearing in our 2D as well as in our 3D here. And then we also have our slices here. So for those who prefer this or to help you orient where your particular 3D model is going through the EM here, we can kind of rotate it around and now you'll see that it's matching here. So we have this part up here and you can see that when I hover, it's highlighting it actually in both sides. So that's kind of nice to give you an idea of an orientation. For this demo though, I am going to hide that, and that's with S for slides or sections. And that's also this radio checkbox down here to show hide that. It's a little easier to kind of see as I show the navigation tools to kind of hide this. To move your axis point and your point of interest around in the 3D and in the 2D, you're going to right click. So that's just going to center you on that point. And that's the same goes for here over in our 2D. To pan in our 2D, you just left click and drag. And as I said at the beginning, left click and drag will rotate you around that point. So to pan in our 3D, it's actually shift left click and drag, and that's going to drag you along the branch itself. Most people use the right click to kind of just jump themselves along a branch rather than panning, but it's up to folks. If you do accidentally do left click, shift and drag here over on our EM, you'll see that it's suddenly getting like striated and kind of messed up. And that's because we're actually actively rotating the EM slide here. And to get that fixed, it's a common user error when you're first starting out, especially if you're like prefer to pan, you're going to hit Z and that's going to snap you back to the axes. And that's because we're in the XY view. For imaging of this data set, the XY view is our preferred image side. The other two views, XZ and YZ, are those composited views where you can see between the slides. We'll show that in a minute and you can see how that looks. But I like to say that if you do accidentally do this, kind of think Control Z to snap you back. So if you accidentally do it, I know I've done it multiple times when I first started out.
Yeah, me too.
Yeah. All right. So speaking of those other views, if we hit spacebar when we hover over a particular view, it will enlarge that full view here. And if we hit spacebar again, it's going to take us to that four view layout here. So we get those other two planes. And you can see why, we prefer to only use the one XY plane rather than the other ones because they're very striated from each of the distances between the thickness of the slices for the cuts of the tissue. Some people will use this if there's a particularly difficult trace, but most people are going to use our side-by-side layout. And to get to that, you can use these little buttons up top here in the upper right corner. And here we can switch back to our side-by-side layout. You can also use them to go to the other like layouts too as well and things like that. So just kind of know that these are here for navigating around that. A lot of people, if you're wanting to take a beautiful screenshot or something, will, enlarge their 3D in order to take a nice one of all the cells of interest for them. Control scroll is going to zoom you in and out, and that's the same for both sides here. So just something to note. And then let's say we want to deselect and reselect our neuron or select something else in our EM here. You're just going to double click to select, and that's going to show up with the cell here. So here we have a nice, I don't know if this is quite bipolar, but it has a merger on it actually, now that I see that. Um, I just randomly clicked here. Oh, hey, that's nice to see. And if we want to deselect and remove that, you're just going to double click to deselect. Something to note, and once again, I forgot to grab an example of this, but I'll talk about the segment IDs in a minute and how we can get the most recent up-to-date version of the segment ID. So each segmentation here, each kind of what's been put into our EM is related to a segment ID. They are made up of many little supervoxels, but these are like all the supervoxels connected together to create our segment. And that creates our full cells. So those segment IDs are going to populate over here in our list on the right. And when I deselect them, you'll notice they no longer have the eye, but they're still available in our list here. So that's kind of nice if you want to, hide something for a minute or, come back to it later, you can do that. If we want to fully remove a cell from the list, you're going to use the star button, or you can do it by shift double clicking in the EM or the 3D. So I can either do this with the star button and remove it, or if I just bring it back, I can just shift double click and now that's removed from my list here. So something just to be aware of. Previously in other versions of Neuroglancer, if you deselected the neuron in the EM, it was lost forever. So this is nice because it does keep a history of it. If you do, just make it hidden rather than completely removing it from your list on your segment IDs. The other thing I'll mention is segment IDs change. So with every edit that happens to a particular segment, that segment ID is updated. We keep a history of that change log. So it is possible that you will open up maybe a link from a while ago or open up an old shared ID and segment ID, and it's going to be the old version. So to make sure that you have the most recent version, especially if you're coming back to something after a while, or maybe it's a cell that's been actively edited, or you want to make sure that maybe somebody already fixed the error in it, you'll just want to deselect and reselect, and that's going to get you the most recent version of that particular neuron. In this case, I didn't have an example of it, but I wonder if I can go and grab one
I just
real quick
posted one
here.
in the chat if you want to use that.
Oh, thank you. That's the one. I knew we had an example and I forgot to save it off to the side. So here we go. Okay. Oh, great. Wonderful. This is a nice one.
I think I may need to upgrade my CPU because it looks like Neuroglancer for my machine is kind of CPU intensive in a way that I'm not used to from WebNosos. So
Hmm.
I've got a 10-year-old, even though at the time it was a pretty good processor, 10 years is a long time in computer world. So I would
Well,
imagine.
I'm actually on a 12-year-old computer, or no,
Okay.
10-year-old as well here, actually 11. And you can adjust that. So in this upper left, right-hand corner here, this settings panel,
Yeah.
you can see here, you can set your GPU memory limit and your system memory limit. I've set it all the way down to 1 and 2. I think the default link sets it at 4 and 8. But this is usually my preferred, especially when I'm streaming this over a Zoom call and it's loading pretty good. So
Right.
that's kind of my best suggestion is to play around with this a little bit. And then, if you're not zooming out constantly and working finely in a certain area, maybe you're, looking at synapses or something, you can change the concurrent chunk request here. And that's all the different tiles that are available in the EM. So here you can see how they all tile in. And so those are kind of like the individual like chunks of data.
Right.
Um, so you can change that down or what up depending on how fast and what you want those to load. Obviously, if you're proofreading a very long wide amacrine cell, you're going to want to make sure that those chunks load up pretty quickly because you're going across the data set quite far. But
Right.
those are some options for you to kind of play with to hopefully make the loading a little bit better,
Right. And there's
especially.
only one resolution of the pre-computed mesh that gets loaded. You don't have a downsampled version that would
Yes.
load faster.
Yes, that's correct.
Right. Okay.
We have like a downsampled version of the EM, and that's specific to the Asia area region, just so that way the EM will load faster for them. And
Right.
it's slightly poorer quality, a little more pixelated, but it will load faster if there's loading issues on that end.
Right. And when you're
Um,
zooming out, you're presumably actually viewing a downsampled version.
yes, you are. Yeah, so you can kind of see that.
Yeah.
Happening when I kind of just zoom out, that it's already doing that. And
Yeah.
you can see also it says like the segmentation won't be loaded. So that means that like it's not loading any of this pink segmentation because I'm so far out in the
Right.
EM.
Right.
So yeah. So that's kind of some helpful, and that's where this adaptive downsampling actually is. And then we have it also with like the prefetching with that too. So there's a variety of kind of options here to help you with loading. And also I'll mention this too, just because we have this little settings menu up, is if you do want to take a particular
Right.
image or picture, you can change the background color, or if you find that black is a little tough for you on something or other, that is possible. Um, so I
That's
do mention that for like screenshot shot
yeah.
purposes.
Right. I do that
And we
all the
have
time.
that with actually we have a little screenshot button right here. So that'll automatically take one and pretty soon download it so you
Where
can
was
have
that?
a
I didn't
little,
see your
it's
oh, up at
up
the top.
here.
I see it. Yeah, got it.
Yeah,
Thanks.
up in the right corner here
Right. Yeah.
next to our
Yeah.
help menu, which I'll mention right now while we're talking about it. First things first, though, I'm just going to reset my screen real quick here because I zoomed out so much, sometimes it has trouble. um, all right. And so we have this little screenshot camera here, so that'll take a little screenshot and you can set it up how you want it and take it. So it's
Nice.
actually really nice, available for kind of taking images there. We also have this help menu, which is next to our settings, which is the question mark. And this is telling you all those
Mhm.
keyboard shortcuts that I
Right.
mentioned for navigation and for moving through different options here. So this is kind of available for you as well as the guides we have that explain each of those kind of actions that are being just written here.
Right.
So that's available. Okay, so let's go back to talking about the segmentation. So here I'm going to bring in a segment ID and I'm going to paste it into this box up top here. And you'll see that I suddenly get a second segment list here. And so we
Hmm.
kind of think of this as our segment search list. And then I will put it down into my active list down here. So if I want this segment to be down in my active list, I can just hit the eyeball button and it will show up. And so we have a nice wide field amacrine here. And you can see it's, not perfect. There's some truncated branches here. I think there's maybe even a merger with maybe one of the branches. So that might tell me that maybe this is out of date, and maybe I want to check before I start proofreading that it is the most recent version of it. So to do that, I'm just going to double click to deselect and reselect, as I mentioned earlier. And you'll see now that that segment ID has been updated. It's decided to do a different shade of purple, um, but it is now a different number here. It's hidden the old version here. So that will still be listed in your active list. Sometimes you might want to just hit the star button and remove it from your list. So then that way you're not accidentally having
Yeah.
it. Some people like to have that just so, let's say I wanted to see the changes between the two of them.
Hmm.
So to do that, I can actually just maybe change the colors here. So let's
I
make,
see.
we'll do purple and yellow here. And we can kind of
Nice.
see where there may have been some changes. So here's some branches here that we can see that were
Uh-huh.
added on.
Right. Right.
So that's actually kind of a little bit of a neat trick to see, what was changed from maybe some initial segmentation or from when you last, opened up that particular cell. Sometimes it's just a minor thing where like somebody's gone in and like, added a, missing a little axonal bouton or something like that.
Spelling. Right.
Um, and I'll mention this too. So you notice I changed it to a custom color here to this light green. Um, and it now has a white box around it. That means that it's going to always stay this light
I see.
green color when I
Fixed
want to
color.
recolor other neurons. So if I hit L it'll start recoloring the other segments. So you'll notice that our starburst neuron here was pink before, and if I hit L again, it'll kind of keep changing those colors around, but it's always going to keep our wide field amacrine cell that one
Right.
set color. So
Right.
this is kind of nice, especially if maybe you're sorting cells and you want to make them all the same color because you've already know what they are, or maybe you're using them as kind of landmarks for finding other particular cells or circuits. You can kind of set them into one set color. So then that way, as you're clicking around in the data set, you don't start getting things that are the same color and getting confused. So that's something that is very uncanny with Neuroglancer. It is absolutely great at selecting things and giving you very similar colors right next to each other, like this two greens. And as we did with when we brought in this old segment ID, it decided it was two shades of purple. I don't know why, but the programming does that. So I like to tell people, if you want to remove this custom set color, let's say we no longer need it to be that set lime green color here, I just right click it and it goes
Okay.
back to being its own color. And because we were looking at the change here, oh, it's just because we have the old segment ID up here. So let me just hide that now.
I think that's also available to you under the render tab up top. I
It
think
is. So the render tab, you can set a fixed color
it's
here,
exactly
and that's
right.
going to fix it for all your segments in this particular segmentation tab. So that's something to be aware of.
Oh,
You
I
can,
see.
yeah. So you can create multiple segmentation tabs. You can like drag them over from another window. So here, let me kind of bring this over so I can actually have multiple ones here. And then if I want, I can make this particular tab that I just dragged over, everything's going to be red,
Right.
you know, and
Right.
then I have this other version of the segmentation where I can have all the multiple colors I want. So that's
Right.
what
But
this
just
does here.
right. So, but to confirm, if you wanted just one cell out of a group of four to
Hmm.
always keep the same color, you want to do it in the segment list by working through
Yes, by
that
like
way. Okay.
selecting the color
Yeah.
you want.
Got it.
So yeah.
Yep.
Hmm. Yeah. Okay, and now that I did that for fixed color, let me change that. There we go. And the nice thing is you can easily switch that off if you accidentally do it. You just select over here on this, and you can also change your opacity. So if you want to have like things a little lighter, especially in your 2D, maybe to see the vesicles better, that kind of thing, you can adjust that so it's a little bit more opaque. I find about 0.3 is perfect.
Yeah.
And you can also change that as well in your 3D here. So this was our 2D that I just messed with. This is the opacity in 3D. Some people like to change it a little bit just so they can see through where there might be potential
Mhm.
synaptic connections. So
Right.
just some ways that that can get adjusted. Um, and you can also change some other things here, like, you know, if you don't want to have that highlighting, maybe it's flashing too much for you, you can turn that off rather than having
Right.
it highlight every time you hover.
Yeah.
So that's in that render tab there. All right. So for what we're looking at here, we've kind of selected a whole bunch of different cells. I still have this search list from that initial segment ID I pasted in up here. If I ever want to get rid of this search list and only have my active list, all I need to do is just remove the search, like from the search list, and I'm back to only having that one active list. So
I'm
just
sorry, what
something
did you
to,
select there to remove that other one?
yeah, here, I'll do it again. So if I put in something in the search list here, all I
Right.
need to do is just remove it from the
Oh, I
search
see.
list. So I
Just
just hit the
took
back
out the
button.
search, took out the
Yeah.
search term. Got it.
Okay. Yeah. And so that's something to note because sometimes it's confusing when you have like, why do I have two segmentation lists here? They're like look the same, and it's just because one is the search list and one is like the active working list, I'd say. So let's get out of these kind of muddy colors we have here. Okay. So that's our segment ID list. And one thing to mention is, you know, obviously you can save the segment IDs and, you know, in your own personal records and things like that. The other thing is that your coordinates are never going to change within the data set. So if you prefer, maybe you just always will save the soma coordinates of your cell. You can always just kind of put that in your soma here and then just copy those coordinates and paste them in maybe next to your segment ID. So then that way, if the segment ID does change, you can go right back to where that set coordinate is. That's also useful if maybe you're saving locations of potential synaptic points or maybe a mitochondria that looks interesting or something like that. You can always paste the coordinates right back in here. These are editable here. So if I just, hold on. If I just hit Control V here and hit Enter, why are we? It'll jump me right back to that spot. So very handy like that. This is also a good time for me to mention in the data set that we do not have the nuclei attached to the soma membranes here. That's on purpose. And once again, here's that uncanny Neuroglancer. I just selected two different. There we go. I had to recolor a few times for it to give me two different colors here for these segments. We are asking people when they do get to edit access that they do not merge the nuclei with the soma membrane here. And that's just because we're trying to keep the nuclei relatively the same segment IDs static, just for ease of purpose of keeping those not dynamically changing, because every time we make an edit, that's going to change the segment ID. So that's kind of the current hope, is not having those merged in with soma membrane.
But what about mitochondria? It looks to me like you've got some mitochondria that aren't included.
Yes, so mitochondria are indeed separate. And let me go back up to our branches and we can kind of go in there for them. Just give me a minute while we load. Let's get that right here. Okay, great. Yeah, so here's a great example. We're asking like proofreaders not to merge these in. We keep them separate, especially because they can be detected separately. And, they're not necessarily affecting us being able to look at the outside of the branch and where it's having its, connections and synaptic areas. Sometimes when proofreading, we have to add them in, maybe to get it across like a gap where it, it's hard to continue it without selecting that particular one, and we have to add it in. But in general, we're keeping those separate. So then that way we can programmatically, I think there's eventually talk of doing that to just identify all the mitochondria and have them as kind of like set lists for people wishing to do research specifically on those different, the variety of those. So, yeah, so that's why we do have gaps.
So don't mess with the nuclei, don't mess with the mitochondria, leave them alone, but anything else that's a broken process or something like that, that's what you would do.
Yeah, exactly. Yeah. So I just kind of tell people, if it's an inner organelle that's completely surrounded like this, then great.
Don't worry about it.
Yeah. But if it's like a true invagination, obviously we're going to connect that in with the actual cell that it belongs to. And that's very commonly a merger point for cells when proofreading, is that an invagination is commonly the computer will have some issues with that. So just something to kind of note. And I was going to say something else, but I've forgotten it now. Oh, well, it'll come back to me.
We'll come back.
All right. So I talked about having the coordinates. There's other ways that you can mark and keep notes for yourself within the Neuroglancer schema here, and that is by adding annotation layers. So if I right click on this plus sign here, it will add an annotation layer for me. If you hit plus, it'll just add a blank layer that you can use to pull in maybe some other, data or skeletons or something like that you're interested in. But if you right click on it, it just creates a new annotation layer for you. This annotation layer is local to your browser, so it's not global. It doesn't go out to everybody or anything like that. So it's a great way to leave notes or kind of keep track or record of where you've been in the data set, that kind of thing. And you can rename any of these. So let's say I want to rename this, my SAC layer here. So all I've done is just double clicked in this section here. And if I click out, you'll notice it's been renamed here. So I can kind of create a few different annotation tabs to, maybe leave some notes in. So let's go back to that SAC real quick here, and we'll leave maybe a couple points for ourselves for annotations. So you'll click on annotations, and to start annotations, you're going to click this dot here, and that says annotate point. And so now I'm in the annotate point here. You can see that in our upper kind of corner here telling me that I'm there. And I can use Control left click to place it either in our 3D here, so you can see that little yellow dot there, or I can place it in our 2D. So this is helpful if maybe we're, wanting to mark particular regions or areas where we think there might be like synaptic connections or something like that. So definitely helpful. And we can leave notes for ourselves. So if we wanted, maybe I could say like, this was the main trunk of the SAC, that kind of thing. Or maybe that it was the soma.
Mhm.
And that kind of, whatever I want. I can even leave like, oh, what's this? Notes for myself to come back to later.
Right.
And the nice thing is you can jump to these. So you can either right click that point, and it's going to jump you right to it. Or you can use the square bracket keys to move through the individual
Yes.
annotations back and forth. So this is really nice if you have a whole bunch of annotations. Maybe you've, pulled the synaptic table that's been, you know, that's in the works to being automatically detected, and maybe you pull in a whole bunch of those points and you want to go through them individually, that kind of thing. Or you've just dropped a whole bunch of spots here, and you just want to move through them and kind of identify each of them and use that to sort it. Um, and the other thing I'll say is, you can delete these, so I can just hit the trash can here. If you left click it, it's just selecting it in the menu here for me to add notes, but it won't jump me to it. So you see the one that I put in the main trunk, it didn't jump me to it, but I have
Right.
to right click it to jump me
Right
to
click
it.
and jump. Okay.
Yeah. As I said at the beginning, if it's not working, try right clicking. And that actually brings me to the other types of annotations you can create. There's bounding boxes. There's also these kind of annotate lines, which are kind of helpful if we are wanting to maybe, put like a pre and post here. So we could like put it in one spot and then say like it's going to this other spot. So you kind of have a line that you can draw between where you think those two points are
Mhm.
connecting,
Mhm.
between maybe two cells for a synapse or something like that. So that's another type of annotation that is sometimes used. And each annotation layer, you can go to the rendering tab and change the color for it. So the second annotation layer that I created, I can change it to hot pink, and then that way, when I want to create some annotations in it, I know that it's a different colored one that I'm
Mhm.
putting in.
Right.
So you can create as many of these. But the minute I close this window, I'm going to lose those local annotation tabs. So how do I save all my work? How do I save all the segments I have selected in my segment ID list? Maybe I'm, leaving for the day and I want to bring my window right back up where I, at last stopped proofreading or something, or looking or examining along a branch. I'm going to use the share button up here. So the share button is going to basically save this current state for you in that link. So I'm going to hit that, and it's copied to my clipboard. And if I were to just paste that right on into my URL
Sure.
here, it's going to,
Reproduce
obviously,
the
I
view.
just did a quick refresh, but it will bring it right back up exactly where I left off in my
Right.
view, all my segment IDs here, and it's going to keep any of my annotation tabs that I've created here.
And all the data that you've entered into those annotation tabs,
Yes, exactly.
annotation list, right?
Yes,
But
yes.
the
You
thing I'm
can
curious.
export these out if you want, as CVs, I believe.
Mhm.
So that is possible, so you don't lose those lists. But this is something for you. And you said a question, and I
Oh, I was just
apologize.
going to ask, so the idea is that for Eyewire 2, there's lots of things that I would be doing with the volume that will instantly be available to anybody else who's working with the same volume, but that's not true of these annotations.
Yes, that's correct.
Essentially, I should only be using the annotations for things that I'm going to be working with locally. And if somebody else in the project wants access to these, because I've identified a bunch of points that might be interesting to them, how would I transfer that? It would be, again, the sharing link, and I would then send them
Yeah,
that
you can
URL and they
share it to
see it.
them. Yeah.
Yeah, got it.
And that can also be exported out. So we've had some folks who will send us like lists of cells they've already ID'd, and then we'll update that into the database. And that's something we are working on, um, some tools to make it so that way people can add global annotations for typing cells. And
Right,
then, and as I think Yulia mentioned at the beginning of the call, that right now we kind of have like a master spreadsheet for like kind of noting like this cell has been, proofread or, it's been typed, that kind of thing. The goal is to make it so that anyone can kind of see that actively within Neuroglancer itself, or be able to query like a table, or very easily to find that. And so that's kind of the next step right now, is getting that into a usable state for folks to be able to, globally have access to it, and everyone's, not needing to redo the same work or anything like that. But
Cool.
yeah, that's kind of the view orientation here. And pending your time, we already are over an hour, so I want to be cognizant of that for folks. I can always show you like how we do cuts and edits if you do desire that at the moment.
Sure.
So
Later. That's
yeah,
fine. Yeah.
great. Yeah. Okay. Any other questions for me regarding just kind of Neuroglancer, or anything like that?
Don't think so.
Yeah, I think we
Um,
covered a lot of it.